session today is part of the project findings that is financed by the JICA, and it's a collaboration between the National Institute of Japan and Fiocruz. And it gives support to the genomic surveillance network of Fiocruz. Professor Ito, we had a very great week with seminars here in Fiocruz and also at the Instituto Couto Maia. And we had also a discussion about the ongoing project with our group in Bahia. This project covers Bahia, Amazonas, Recife, Ceará, Pernambuco, Ceará, and Rio de Janeiro. So Professor Ito has a large work in computational science. He has background in information system and technology, and he has research in mathematical modeling of virus outbreak, like SARS-CoV-2, influenza, and other viruses also. And he is part of the University of Hokkaido from the Zoonotic Center there. So we're gonna present his results in predicting the trajectory and a replacement of SARS-CoV-2 variants. He gave also a seminar last week in Rio de Janeiro about this bioinformatics tool and all the algorithms behind. That was also very fruitful. So I would like to invite Professor Ito to present his data. I just want to mention that we have also here Professor Itamura representing the National Institute of Japan, and also Tominaga-san as the JICA coordinator of the project. Thank you. Professor Ito, please.
Okay, so thank you for your kind introduction, Ricardo-san. So my name is Kimihito Ito from Hokkaido University in Japan. So let me first introduce about my institute and my country and my city or something like that before the introduction of the research topics. So for this one, so this is the picture showing Japan and Brazil. So you can see that Japan is a very small country, and then population is it's also smaller than Brazil. But because of the area is very small, the density, population density is more than 10 times higher than Brazil. Okay, so and then its location is Brazil is southern hemisphere, and then Japan is northern hemisphere, and the time difference is 12 hours. So which means it's completely opposite side of Brazil, and then so I came here. Arrived at Rio de Janeiro last week. I flew from Sapporo to Osaka, and then Osaka to Dubai, and then Dubai to Rio de Janeiro. But basically, we can go any direction because it's the opposite side of the Earth. So then any direction would be possible. Okay, so and then I belong to Hokkaido University, and then Hokkaido University is located at Sapporo, a city called Sapporo. Maybe you have heard of this city from the beer, Sapporo beer or something like this. Good beer, thank you. So this Sapporo beer was established by the Meiji government. Okay, and then so at the time, end of the Edo period, Japanese government, new government, want to westernize everything. Okay, so system and the education system, and then government system, and so industry, everything to westernize. And then Sapporo beer was established by the government, and then it was the first Japanese beer brewery. Okay, and then they invited some engineers or technicians from Germany, and then they built Sapporo beer in here. Okay, and then area is something like this, and then it's a northern part of Japan. It's almost Russia. It's very cold, and then you can see this kind of amount of snow in the winter. Okay, so it's a higher or taller than the height of car. You can see, and this is where the city where I live. Okay, so the city was new, so it is established in 1868 or something like this around here. And then before that time, this Hokkaido island is almost all forest. It's no Japanese were very few Japanese were lived there, and then Japanese government decided to have a capital of Hokkaido prefecture at the Sapporo. Okay, and then in 1868. Okay, and then 1876, and then Japanese government established Sapporo Agriculture College, which is a former name of Hokkaido University. And then, so this is the university is responsible for the research and the education of agriculture. So, and then it founded in 1876, and then all professors were invited from Western countries such as United States, and then countries from in Europe or something. And then our educational philosophy is a frontier spirit, because it's a frontier of Japan. So and then it's almost all was forest, and then developed some new land for Japanese. Of course, there were indigenous people were called Ainu, were lived there, and then it's something like a situation similar to United States or something like this. Also from Brazil, South American countries. Okay, so then frontier spirit is one philosophy, and the second one is global perspective, because it was already international educational environment from the beginning, because all professors are non-Japanese, and then maybe classes would be taught by the English, and then so that is a global second educational philosophy. And then all around education, it's not only some study, but also from leadership or something like that one. So it's all around education, and then practical learning, because it started with the agriculture university, and then so practice is implementation, or to implementation of the research result would be was very important. Our university thinks the implementation of some result, our research results to the society is, and the contribution to the society is important for our university. And then students, we have 11,000 undergraduate students and 6,000 undergraduate students. Okay, so, and then Hokkaido University established Research Center for Zoonosis Control in 2005. So at that time, so many emerging infectious disease, it's also now it's problem for society. And then most emerging infectious diseases originate from animals, such as wild animals or livestock. And then disease in animal or livestock and wild animals also it's responsive. Veterinary medicine is responsible for the infectious disease in livestock and wild animals. And then medicine is responsible for infectious disease in humans. But if we think about zoonosis, which transmit from animals to human, it's a transboundary area. So there was no responsible education system about this one, because veterinary medicine teach about infectious disease in animals, and while medicine teach infectious disease in humans. So no research organization which focus on this zoonosis was there was no such a organization, so or research faculty. So then Hokkaido University decided to have a research center specific to zoonotic infectious diseases. So it was designated at the WHO Collaborating Center for Zoonosis Control in 2011. Okay, so let me introduce myself. So I'm a professor at the Division of Bioinformatics at the International Institute for Zoonosis Control and Hokkaido University. So I belong to that institute. And then I graduated from I got a PhD in the engineering. So before I moved to the Research Center for Zoonosis Control, I did undergraduate and graduate PhD study, undergraduate and master course and PhD study, and postdoc and assistant professor in Graduate School of Information Science and Technology in Hokkaido University. So at that time, my research topic was machine learning and data mining. So I'm working on some basic research about machine learning. But I was interested in applying this technique to some actual problem, such as health problem or life science or something like that. And then when Hokkaido University established a research center for zoonosis control, I moved from information science technology to this newly built established institute in 2005. After that, I was I have been working on the prediction of the evolution of influenza A viruses. And then when we had SARS-CoV-2, I applied this method to SARS-CoV-2. And then today I'm going to talk about this the especially the prediction method for variant replacement of SARS-CoV-2 viruses. Okay, so emerging infectious diseases in pathogens originate from wild animals or livestock. And then human to human transmission of virus cause pandemic if it adapts to the human population and then efficiently transmit between humans. And then, so if we have pandemic, sometimes we have pandemic because nobody has immunity to the new viruses because it originates from animals or livestock. No, maybe in the most case, no immunity to these viruses. And then after the pandemic, there are two scenarios. One is after everyone is having immunity to this pandemic virus, they stop the circulation because of the herd immunity. Second scenario, which is the most case, is this one, is that they continue to circulate in human population, and then and by evading immunity to the immunity acquired from previous infection. So when it's random mutations are important for the virus to evade immunity, and then so natural selection of the variants will select the variants, and then new variants emerged again and again, and then it keeps circulating in human population. That is the second scenario. So this is the examples are of course SARS-CoV-2. So it have a pandemic, and after everyone got immunity, it still circulate because they mutate, and then viruses which can infect to the people who have infected before is selected in the population. And another example is influenza. Influenza is also zoonotic disease, and then we have a seasonal flu every year. But and these are also from the results of the selection of the variants. Okay, so let me talk about the about selection of the. Let's think about selection of the viruses. Okay, so these are structures of the influenza and SARS-CoV-2 viruses, and then of course they have a genome inside their virions. So these are influenza viruses, and then it is 8 segmented RNA located inside the virions, and these are SARS-CoV-2 is a single strand genome is located. And then replication of RNA virus it needs a living cell, and then so it's even if we give some nutrition or everything they need, virus cannot replicate. virus needs a living cell to replicate. So this is just an illustration of the virus replication. So basically, virus has their own RNA or DNA or something like this, and then when virus infect to a cell, they release their genome to the into the cell, and then cell is of course have own DNA, and then its activity is transcribe this DNA to make messenger RNA, and the messenger RNA is translated to proteins, and then our cells is producing proteins which is necessary for our activities. But in the case of the virus, virus hijack this system and just release their produce messenger RNA, which encodes the proteins, amino acid sequence of the virus proteins. So, and then our cell starts to produce viral proteins instead of human proteins, and then we will have all proteins necessary to all virus proteins. And then one thing host cell can do not do is the replication of viral RNA. So in the case of influenza or some viruses, they have some polymerase inside their virions, and then they create a copy of their own RNA, and then if when every component of virus is prepared, then it's assembled at cell surface, and then bud from the cell, and then released from the cell. So this is the process of the viral replication, and then the and then its process is very similar to chain letters. So this is a letters called letters of happiness. Maybe younger people don't know about letters of happiness, but maybe some older people than me may know about it and have received this. So now, currently, email chain letters comes by electrically. Okay, so it's about email or something like this. Okay, so once I have received such kind of chain letter by email, so there are many dogs in public health authority in Hokkaido, and then they have 100. One big breeder was bankrupt, and then hundreds of dogs, baby dogs, are kept in the public health center. And then 2 weeks after 2 weeks, they will be killed. And then please, if you receive this email, please distribute this email to others who may become the new owner of these puppies or something like this. I received such one, and then I believe this was such kind of very tricky or one, and I sent this to some of my friends, and then he said it is chain. Okay, so it is not true. It was not true. So now such kind of chain mail is comes from the through electrical way. But 50 or 30, 40 years ago, there is of course there was no email system, and then it came by the actual physical letter like this, and then we sender and the receiver needed to use handwriting or typewriter to create it. And then this version, older version of chain letter is much more strong because they can mutate. So in the email, it's just forward to others, and then do not have chance. But this one is copied by hand. Okay, so, and then sometimes they introduce some error, and then sometimes they introduce some new sentence, which is scary one or something like this. So the letter which get some grammatical error, for instance, have a lower probability for receiver to copy another one. But the letter which acquire very some good phrase, then they have the probability for receiver to make another copy becomes higher. So during the circulation, only good letters are selected, okay, and then bad letter is not copied, and then only good letters can copy. And then during circulation, the letter obtains better and better phrases, and then it becomes finally this kind of very sophisticated letter will be produced. So it's a very similar to virus evolution or something like evolution. So letters cannot replicate by themselves, and the letters don't know anything about what kind of phrase is good. Letter does not have any knowledge. Just but letter can acquire very good phrases. Letter does not know anything about English grammar or something like this, but it can evolve, and that is very similar to virus. Virus is a DNA version of this one. Just they just make a random copy or something like this, and then good letter, a good DNA or RNA is selected. Okay, so then this is example of example of nucleotide sequences of viruses. So this is the nucleotide sequences of H1N1 in HA sequences of H3N2, which caused the Hong Kong flu pandemic in 1968. So, and then 3 years later, this virus acquired these nucleotide changes, shown in yellow, and then 40 years later, these blue are so these mutations from compared to the 1968. Okay. So so this is the mechanism of the virus evolution. So replication induces copy errors in RNA or DNA, and a large variety of descendants are produced, and the virus adapting to the environment are selected. So this is just some phenomena similar to the chain letters. So natural selection is it's a fundamental role. Okay? So then it can act to letters, act to viruses, or something like this. This is just a fundamental role of evolution. It's not, and then virus evolution is one of that, one of the good, a good example of natural selection, and the chain letter is also very easy example to understand. And of course, ourselves has evolved with this mechanism. Okay, so let's talk about the virus evolution or uh, in pandemic, in a pandemic. So there are two different disciplines which studies about the pandemic. So one is epidemiology. So epidemiological analysis basically look at the number of infections. Okay? And then, so people in epidemiology or researchers in epidemiology analyze these numbers. So, and then to calculate basic reproduction number and or effective reproduction number. Sometimes they use mathematical model, SIR model, or renewal model. And then on the other hand, and there are another discipline handling epidemic, which is genomic science. Okay, and then it's genomics scientists, molecular genetics scientists basically analyze genetic sequences of viruses, conducting, for example, phylogenetic analysis or coalescence theory or population genetics or natural selection. Okay, so these are different, two different disciplines before SARS-CoV-2, and then but they basically focus on the same event, which is pandemic. One is epidemiology, and the other is genetic analysis. So, but this research looks at the same phenomena, and then we think these two areas should be combined to collaborate to analyze the phenomena of pandemic. Okay, so and then to conduct mathematical modeling or bioinformatics study, we need large data. Okay, and then before the SARS-CoV-2 pandemic, I'm also conducting a study of influenza A viruses. So in the case of influenza viruses before SARS-CoV-2 pandemic, weekly number of infections are reported by WHO or countries. And then as of September 20, last week, we have 500,000 sequences stored in GSED database. And then it includes influenza virus sequences of humans or birds and pigs and other animals. So if we look at the whole human viruses, and then the number is much lower. Okay, so in the case of the H1N1 PDM pandemic 2009 strain, it has 100,000 sequences. So we have this in 2009, so it is 15 years ago. So 100,000 sequences in 15 years. This is the situation of influenza viruses. But if we look at SARS-CoV-2, we have daily number of infections and deaths and WHO and by the countries, and then sequences of 16 million strains in just 4 years. Okay, so 100,000 strain sequences in 15 years. This was the previous situation, but now in the case of SARS-CoV-2, we have 16 million sequences with just only in 4 years. So we have in other words, we have enough number of epidemiological data, or enough number of sequence data in the case of SARS-CoV-2. And then I applied mathematical modeling studies to analyze both of these data. So, and then today I'm going to talk about the study I conducted during SARS-CoV-2. So this was the collaboration between Hiroshi Nishiura, who is a very famous epidemiologist in Japan, and he is actually famous in the world. And then he's younger than me, and but he has maybe about 200 papers already, and then he's one of the leading scientists in infectious disease epidemiology. And then his area is epidemiology. My area is virus evolution. And then we collaborate together. We analyze the SARS-CoV-2 sequence and the epidemiological data together and in 2021. So this is the first example I'm talking about today. It's about prediction of the dominance in the Delta variant. So let's go back, look back in the June situation in June 2021. Okay, it's 3 years ago. Okay, so in June 2021, the Delta variant showed high transmissibility in India and the United Kingdom. And at that time, Tokyo Olympic Games are scheduled to start from July 23rd. Okay. So replacement of Alpha by the Delta was posing public health threat in Japan. But we have only 1 month. And an important question as of June 2021 was how transmissible the Delta was compared to Alpha. And then when the Alpha be replaced by the Delta. So this was the question. But and then nobody knows about this at that time. So we did some mathematical modeling study about this one. And then, so the mathematical modeling study, I use effective reproduction number, and then especially relative effective reproduction number. So let me talk effective reproduction number first, and then I will talk about what is relative reproduction number in the next slide or later. And then effective reproduction number is defined as the average number of people an infected individual at time t could be expected to infect. So this is the definition. So when we have one, okay, so and then this is defined by, calculated by this formula. IT is the current or number of infections, something like this. And then J is the generation time distribution. So how long does it take from primary infection to the secondary infection? So if this is, let's say this is 4 days, then it's let's okay, so let's 4 days or something like this. So this is the generation time, and then using the generation time, we can calculate the relative effective reproduction number by this formula. So this IT is the current number of new infections, and then this is the numerator of this formula, and the denominator is the generation time probability of generation time multiplied by the number of infections in J days ago. Probability of generation time is J multiplied by number of infection in J days ago. For instance, if let's assume that generation time is 4 days. Okay, so then in that case, effective reproduction number is calculated by the number of new cases of today divided by the number of new cases in 4 days ago. Okay, so then 4 days ago, how many people were infected? And then today, how many people with infected? If we divide this one, we can calculate how many secondary infections we have. So then this is the formula. So basically, the numerator is the current number of infections. The denominator is the expected number of infection in previous generation. Okay, and then relatively, of course, if effective reproduction number is, if it is 1, then number of new cases stays the same, because one person infects the other another one person. It is not going up and not going down. It's same number of infection continues if effective reproduction number is 1. And then if effective reproduction number is more than 1 or greater than 1, then we will face exponential growth of the infection. So if the effective reproduction number is 2, then 1 becomes 2, and the 2 becomes 4, and the 4 becomes 8, and something like this. And then effective reproduction number is less than 1, then epidemics start to reduce. Okay, so effective reproduction number is, for instance, 0.5, then 8 becomes 4 in next generation, and then 4 becomes 2, and the 2 becomes 1, and the 1 becomes 0 or 0.5 or something like that. So effective reproduction number is very good, important index to measure the current situation of outbreak. So the control to measure to reduce the effective number below 0 becomes important. Okay. So using this effective reproduction number, we defined a new concept called relative reproduction number. I am calling relative instantaneous reproduction number in my paper, but in this presentation I just call it relative reproduction number, relative effective reproduction number. So relative reproduction number is ratio of effective reproduction number of two variants. Okay, so for instance, so let's assume large A is mutant virus, and then small a is wild type viruses, and then effective reproduction number of mutant virus divided by effective reproduction number of wild type virus is defined as relative reproduction number. So, for instance, and then if we think this K relative reproduction number is 2, it means that the mutant virus large A is 2 times more transmissible than wild type viruses. And then we assume that relative reproduction number is constant over time. Okay. So let's suppose the relative reproduction number is 2. Okay. So then if let's suppose this is 2. Okay. 2. Okay. So then when in this case when if effective reproduction number of the wild type is pointed to 1, okay, and then effective reproduction number of mutant virus is 2. Also, it's 2. Oh, it's 2. Okay, and then when the effective reproduction number of mutant virus becomes, for instance, 1, okay, then its effective reproduction number of wild type virus should be 0.5 or something like this. So it means that mutant virus is always 2 times more transmissible than wild type virus. That is the definition. Okay. So then, by defining this one, we can simplify by just assuming this one. We can calculate the trajectory or frequency of mutant virus with this simple formula. So derivation of this formula is written in my paper. So if you are interested in this derivation, then please look at the paper. Okay. So please note that this formula does not contain effective reproduction number or number of infections. So just knowing K and the generation time, we can calculate the trajectory of the virus replacement, and then in actual by doing the reversely, we can estimate K from the observation. So this formula calculates the frequency from K. But what we can observe is frequency, right? So by using maximum likelihood estimation or fitting, we can estimate relative reproduction number K from the observation. Okay. So that is what I was doing during my during the COVID-19 epidemic. So this is the first result I would like to introduce. So this is these graph plot shows the virus replacement in Japan from in 2021. So x-axis, of course, it represents time, and the y-axis represents frequency of mutant viruses. Okay, so the yellow one is viruses from the original Wuhan strain. We have original Wuhan strain circulating in Japan around December in 2020. But this purple one, which is R1 strain, which is the Japanese original mutant of Wuhan one, start to replace Wuhan strain like this. But around here, we have arrival of alpha variant. And then alpha starts to replace the original Wuhan strain, the R1 strain in Japan, and something like this. And then we can see small increase of Delta variants around here, like this one, green. Okay, we cannot see this one. So we have seen small increase of Delta variants like this. So from these data, we calculated the relative reproduction number of R1 and the alpha and the Delta compared to original Wuhan strain. And then from this analysis, we found that R1 strain is 1.25 times more transmissible than original Wuhan strain. And then alpha is 1.44 times more transmissible than others, than original Wuhan strain. And then delta is 1.95 more times more transmissible than Wuhan strain. And then important thing was the delta was predicted to take over alpha before Olympic Games around here. And then we reported this one result to the government. Okay. And then in the advisory board of Ministry of advisory board meeting of Ministry of Health and Labor and Welfare. So due to the replacement of the delta, replacement of alpha by delta, it is expected to have relative reproduction number increase. So then this one is a prediction results using data up to June 14. And then these are results of the these are actual observations after predictions, and then it is you can see that the prediction was almost perfectly matched the prediction. And then we applied this same method to the Omicron variant, and then we found that Omicron variant is 3.19 more times more transmissible than Delta. And then this result was published in CDC and then CNN. And then because these are one of the first reports stating that how Omicron is very Omicron transmissibility is very high. Okay, so these are the result of the prediction. And then these are actual observations after the prediction. So we can see that the prediction was almost perfect until 50%, but we can see some gaps between predictions and observations. So we analyze why this asymmetry is observed, and then we end up with the conclusion that the generation time of Omicron is shorter than Delta. So if we assume that generation time of Omicron is shorter, then we can perfectly match, we can get the trajectory which perfectly match this transition trajectory. Okay, so from this result, mean generation time of Omicron BA1 was 1 point, or about half of that of Delta. Okay, so this is third result I'm going to introduce is about how much data will be needed to estimate to predict the future. So I did some prediction at the 5% and the 10% and the 15% and something like this, and compare the prediction result with the actual observation. So this is the result of the prediction made at the new Delta variant was 5%. So these are predicted trajectory and these are observed number. So we can see that the small, although this is within 95% confidence, almost around the 95 confidence interval curves, but we can see that some gaps between prediction and actual observation. So 5% is not enough. Okay, and then so this is a prediction made when Delta variant or new variant becomes reached 10%. Then we have some improvement compared to the prediction at 5%, but still have some gap between prediction and observation. And then this is 15%, then we have almost perfect result. So then after that, this one, we can see this 20%, 50%, 30%, and 35, 40, and 50%. It's almost perfect prediction. Okay, so these are prediction of the date which new variant reach the 90%. So this is the last observation date, and these are predicted date of when new variants reach the 90%, and then this is the actual date when new variant reached 90%. So using this is 10% prediction is not was not very accurate, but and 10% prediction was not also very accurate. But after 15%, we can have very accurate prediction results. So once a new variant, the relative frequency reached 15%, the date when the relative frequency of new variants will reach 90% was predicted with maximum absolute prediction error of 3 days. Okay, so and then this is the last result I introduced today, and then which was collaborated by collaboration with Fiocruz. So and then I received an email from Gonzalo, and then I started to analyze the uh, frequency of Delta and Omicron in Brazil. And then combined with other results, we published that paper. And this is the paper, the result. I was responsible for that paper. This is the frequency of the gamma, and this is the frequency of delta, and this is the frequency of Omicron in Amazonas area. And then by analyzing this frequency by our method, the relative reproduction number of gamma and Omicron BA1 with respect to delta was estimated to be 76%, 0.76 and 3.25 respectively. Okay, so this is a summary of my talk. It's not possible to stop the replacement of the variant. So variant is selected by natural selection. So there is no way to stop the replacement. Okay. And because it's a always new variant is selected by the natural selection. This is the law of the general law. We cannot stop the evolution. The future trajectory of variant replacement can be predicted. However, accurate prediction were expected after the new variant reached around 15% in frequency, but it is just about 1 month before the replacement. So for public health policymakers may have only short time to prepare control measures for the increase in viral transmissibility, and a quick decision-making process is needed to take advantage of the prediction. Okay, thank you very much for your attendance.
Thank you, Professor Kimihito. Questions? I think you have time for 1 or 2 questions.
I just want to make a remark. We discussed it a lot in the beginning of the week and the other things. I just want to thank you for the whole presentation, because you come really from the beginning of viruses and evolutions, and I really like the concepts that we brought about pandemic, and also the explanation of the virus evolution of the letter. It was really nice to become a material that we can use with students here. If I had this class, maybe long time ago, I would be in another level of evolution for sure. Thank you.
Mariuda, our director.
Congratulations for your talk. It was very nice to stay here and listen to everything that you are doing. And I want to know, it's the only curiosity about if you can using your model to other situations outside SARS-CoV-2 and the other organisms outside virus. If you think you can make this, if it's possible.
Yeah, thank you very much for that very important question. So basically, we can apply this method to other viruses or other infectious diseases. But we need to think about this assumption. Okay, so so assumption is that the relative reproduction number is constant. So as long as this assumptions hold, then we can apply this one. Okay. So for instance, okay. So let's think about the lockdown or something like this. Then lockdown or social distancing. So then lockdown affects both strains in SARS-CoV-2 or something like this. It is if the reproduction number of one strain go down by half, and the reproduction number of other strains go down by half. So this is all. So this ratio is always constant. So that in such a case we can apply this method. So for instance, if we think about vector-borne disease, which use the same vector, okay, so the same mosquito or something like this, then maybe we can assume such a situation. So because vector-borne diseases number of vectors are very it affects the reproduction numbers of vector-borne diseases. So if some diseases use the same vector, then I think we can assume this assumption and it can work. But we cannot apply this one when vector is different or something like this. So because only if we if one vector goes down, then if other vector does not stay same, then relative reproduction number of one virus go down, but others stay same. Then we cannot assume this one. The another situation we cannot apply is that some situation where vaccination and the vaccination reduce effective reproduction number of one virus or one strain. So for instance, let's think about the influenza viruses, and then if one vaccine, in case of influenza, we have H1N1, H3N2, and B, it's mixed together. And then if one vaccine works very well, and then one vaccine for one subtype works very well, and then vaccine for another subtype was not very doesn't good, wasn't not very good, then this balance or this formula does not hold anymore. In such case, we cannot predict.
Okay. Any question? I have one last question. I was wondering if you can apply this model for example, for example, for bacteria or the cases of bacteria, for example, pneumococcal, when you have a serotype replacement from the bacteria, or the reproduction number will be so low because the serotype is replaced in a year, for example, and then you cannot apply this model. I was wondering if you can apply not only for virus, but for example, for bacteria that, for example, pneumococcals or something like this.
I haven't think about one, but I think it's applicable. I think. Even if as long, but maybe difficulty would be that long generation time.
Yes,
so then
because the strain changes every year, for example, or every 2 years, so it's different from virus. But I was wondering about to apply this model for other for
it's
serotype replacement.
Very interesting. I think that maybe we can discuss about that. So if you're interested in
Okay.
future, maybe you and then I have applied this method in You
Measles.
see, it's a it's a viruses, but it's a longer longer generation time. But it's just 10 days. It's compared to the
Bacteria, yes.
But in case of, for instance, tuberculosis or something like this, I think it's too much, too long.
Yes, yes, that's the question.
But it's one generation time is months.
Yes.
So it would be possible.
Okay.
But I don't know. It's very interesting question. Thank you
Okay.
very much.
Thank you very much. Thank you for a great talk. And I will close the session.